It's a great pleasure to be here with you. And thank you to the organizers. Thank you to the GIP-TRIAD and Dr. Fabien Lombardo for inviting me. I thought today I would talk about algae as a biotechnology tool to combine wastewater remediation, biofuel production or hydrogen, and biostimulant generation. So those are the themes that we are studying in our lab. And I'm going to present a little bit of everything in a general manner. Really the project that we have right now ongoing. So before that, I want to introduce a little bit the world of algae, because I think when we talk about algae, we immediately think about macroalgae, the algae we found in the ocean, on the beach, and also the algae that we eat. And I know in Japan, sushis and other dishes are well famous to use macroalgae. But here I want to focus on microalgae that are very small eukaryotic microorganisms, photosynthetic, that usually range between 1 to 7 micron, and that we only see in microscopes. And you can see here there's quite a wide range of diversity. Here I'm only showing green algae, but of course we have red, brown, and other type of microalgae. And in our lab, we really work only with those small green microalgae. And you can find them in ponds, in swimming pools. They invade a bit of everything. And here in Spain, we also cook with microalgae. And there's a famous chef, Angel Leon, that is developing dishes with this with plankton, and is quite good, and it's becoming really famous. So here in our lab, we work with a green algae called Chlamydomonas reinhardtii, or clammy, we call it more commonly. It's a small green algae that has 2 flagella and a chloroplast that his size is 7 micron. And it's a model organism because we have the genome. It's been sequenced a few years back now, and we've been using it for many studies related to photosynthesis, flagella, motility, biofuel production. We have genetic tools, so really we can do many things with this algae. And it looks like this under the microscopes. You can see that this round cells is able to move around thanks to its flagella. And of course, the chloroplast is really big. And so we kind of do all our studies with its organisms. So microalgae are very easy to grow. You only need sunlight, CO2 and water. They are able to uptake mineral nutrients and fix CO2 from the atmosphere in order to grow. Their replication time is around 7 hours. And so it makes this, this organism very easy to cultivate and to investigate in general. You can see here on the left a photo from the University of Hawaii, who has been cultivating algae for decades now, and have a really big installation cultivating green but also red and brown algae in large scale, actually in pool. And on the right, you can see tanks that are developed and that we are using nowadays to cultivate in really large scale. But we have also new types of photobioreactors indoors and outdoors that allows us to to really cultivate microalgae in a huge quantity. So we know how to do that. Of course, depending on the algae that you are using, the conditions and also the way to cultivate them is going to be a little bit different. And that's what each lab is investigating. Because thanks to the biomass obtained from this microalgae, you can make many products. And for example, the end products from biomass can be used for cosmetics, drugs, supplements, vitamins, food, fertilizers, animal feed, any kind of raw material industries, bioplastics, and biofuel. So pretty much anything you want to make, anything you name it, we can do it with the biomass of microalgae, because it contains a lot of lipids, carbohydrates, proteins. So it's very useful in terms of raw material. And of course, it's eco-friendly. So you can make a wide range of bioproducts that are, let's say, renewable in the sense that you can reuse it or recycle it. So it's quite convenient. And attached to that comes the biorefinery concept. Since with algae or microalgae in general, we are able to use CO2, the biorefinery concept is very useful to fix CO2 released by any kind of industry, and then also remediate wastewater that is released during, for example, dairy industries or olive oil industries. So you can combine sunshine, CO2 released from polluting industries, and wastewaters released also from them, and grew your algae. And that's the research that we usually do at the laboratory scale. We go from fundamental research to large scale, and then to application. So basically, once we have our biomass, we harvest it, we want to break it, fractionate it to obtain protein, lipids, or carbohydrates, in order to choose the corresponding application and see what's relevant depending on the products we obtain from the biomass, knowing that each biomass will be different depending on the microalgae you use, and of course depending on the wastewater you use and the conditions under where the microalgae were grown. So in our lab, what we do is exactly that. We grow algae in different conditions, but we also combine them with bacteria. We try to really improve those conditions in order to obtain remediation processes that are better and also more biomass. And that will lead to the same application, but sometimes with a specific application in mind, because the biomass obtained is richer in lipids, for example, or richer in proteins. But overall, we have the same categories, which is biofuel, biofertilizers, and feed and food. So we have different kind of ways basically to maximize this biomass. And here I'm going to talk about a specific project that we have in the lab, which was dedicated to water bioremediation using Algae-bacteria consortia for rural areas. So this project was in collaboration with 4 partners, 2 from France, 1 from Morocco, and 1 from Spain. And the idea behind that was that we would be using rural water from France in this case that we would identify specific microalgae bacteria consortium able to remediate this wastewater. Then we would, of course, optimize the bioremediation process and develop new harvesting techniques in order to get a little bit cleaner water. And from the biomass obtained, we would do agronomical valorization. In this case, trying to get biostimulants for plant growth, and try to do an overall economical sociological study about the overall process impacts. So what we did here in Spain is use our favorite algae, Chlamydomonas, and we tested it with different bacterial strain that we had in our lab. Here, 4 different types. That means that they have different metabolism, and of course they require different energy sources. But we tested them so together, and we were looking for a successful consortium. And what we meant by a successful consortium here is that they are able to grow better when together, and they are able to consume nitrogen. Nitrogen being one of the most contaminants in residual water. So typically what we did is we combine the 2 microorganisms in a flask. You can see here in the middle. And we had our control, which were Chlamydomonas, the algae alone, and the bacteria alone. We grew it in different ammonium source or nitrogen sources, and we measure ammonium and nitrate concentration to look at the nitrogen consumption overall of our 3 flasks. And then we use different type of carbon sources to look at the growth parameter. And that was done measuring the chlorophyll, the optical density, the size pellet, which is dry biomass, counting cells, and so on. So from this test, we were able to discover one great partner, which is a pink bacteria, which gave the best results, meaning that using a specific carbon source here, which was acetate, ethanol, and methanol, the growth outpassed the other consortium we tested. And when we looked at nitrogen consumption, again, the results were much better when we used Chlamydomonas with this pink bacteria. So typically, we grew our cells in different flasks, and we looked at the growth parameter after 4 days and 11 days. And you can see that the co-culture or the consortium growth is much improved when we combined Chlamydomonas and the pink bacteria. So we obtained a really dark green, and then this was confirmed by chlorophyll measurements. Here you can see on the left, the consortia in yellow is reaching around 50 microgram of chlorophyll per milliliter after 7 days. And it's almost 5 times more than our control algae. And if you look at the optical density on the right-hand side, again, the consortia in yellow is reaching 8, which is almost 8 times more than our controls, bacteria and algae. Next, we also looked at the cell and the pellet, which is correlating the results of our dry weight measurement. You can see on the right-hand side that we obtain almost 40 grams of biomass for the consortia, which is almost 40 times more than our controls. And finally, we looked at nitrogen consumption. So on the left, you can see that the ammonium level is reaching almost zero after 4 days of co-culture when using our consortium. So it's working pretty well. The consortia is able to uptake ammonia from the media. And if you look on the right-hand side, again, the nitrate-nitrite ratio is going down after 7 days, reaching almost zero. So it's much slower, but again, the consortia is able to uptake ammonium and nitrate and nitrite much better than our control algae and our control bacteria. So we have here a successful consortia. Since the consortia is able to grow more and to uptake ammonia or consume nitrogen in general, let's say, and in synthetic media. So until now, we were doing tests in the lab, at the lab scale. And so we wanted to try our consortia in real wastewater and really look at the suitability. So in this case, we received wastewater from our partner in France, Saint-Paul-les-Durance, and we grew our consortia in this wastewater. But here you can see that the results were not so good. In yellow, the consortia is reaching similar level as our control algae and bacteria. Regarding risk of the we thought we improved that. Well, our pink bacteria is actually a methanogen. We should add in our water. 7 days, we observed a big difference, and after 11 days, here we can see that the consortia is really happy and it's now growing really well compared to our parents or control organism. You can see the photo below the graph, where the consortia is really green, so the algae is very happy, whereas the control Chlamydomonas is now yellow, so it's almost dead. So methanol is working very well, and we thought, well, maybe it's not great to use methanol in wastewater, but in fact, methanol is found in many types of wastewater, and also it's used in remediation in general as a process to speed up decontamination. So we measured again the dry weight to confirm what we were seeing, and we see that the consortia is able to grow 4 times more than the rest of the control, and that is also correlated with the pellet that we see below, where you can see a mix of green and red here. So this consortia is working in our rural wastewater, and the next step is now to upscale. So our partners in France installed 3 times 100 liter raceways. You can see here they have a paddle wheel, and they are able to grow the algae in open raceway, or they can also close it. They try to optimize or this setup automatically with a pump, a data logging and pH control, and they started growing our consortia, our winning consortia, and trying to get biomass. But of course, it's not that straightforward, because you cannot just put your cells directly in 100 liter of wastewater. You have to adapt them, first of all, in a 1 liter bioreactor in synthetic media to make sure that they are happy, that you can observe your bacteria in your algae growing together. And you have to do different phases in order for the the batch to be happy. So you start with your synthetic media, then you use filtered wastewater, and you can see here in the second phase, it's going down, it's almost crashing down, but it's normal. The cells need to adapt to a new media that is less rich than the synthetic media that we are using. And then once it's almost stable, you can add raw wastewater in the final phase. So it takes almost 30 days before you can grow your algae consortia, Algae-bacteria consortia in raw or real wastewater. From that, you need to grow it until you get sufficient biomass, and you need to harvest it. So to harvest this biomass, there's different ways, and our partners were trying to see if algae is able to autoflocculate. That means on its own, it will aggregate, and so you don't need to centrifuge it, and you will get basically all your consortia at the bottom of a flask. And this was not the case really for our consortia. We know that Chlamydomonas doesn't flocculate itself, but we were hoping that maybe with the bacteria, because sometimes that happens, the consortia together will be able to flocculate. So they went for a second type of techniques. They tried to develop a biofilm technique using their Raceway and their paddle wheel. They put in place a film where the algae consortia, Algae-bacteria consortia is able to attach and then is able to mature on, along the time and disperse on its own. So once the biomass is attached, the weight of the consortia will lead to detachment on itself. So we don't need to centrifuge, we don't need to do anything. You just collect at the bottom of this biofilm that the biomass obtain. And that's really helpful in terms of biotechnology. But then, of course, it's full of water, so you need to dewater this biomass in order to get a usable biomass for subsequent application. So what was done next is to freeze the biomass obtained previously and then lyophilize it in order to send it to our next partners. So the biomass lyophilized was sent to Morocco, where they were in charge of assessing the impact as biostimulant on tomato plant growth. And so that's exactly what they did. The question was really, can we improve the growth of tomato plants using the biomass from our successful consortia? So before to do that, of course, you need to optimize algae biomass hydrolysis. You cannot just use it directly, you need to fractionate it. So they did different type of pretreatments using acid, basic, or enzymatic pretreatments to characterize the biomass in terms of carbohydrate, protein, and lipids. And then they hydrolyze this biomass that can be then used directly on the plants. And the results show that, well, a specific treatment here based on hydrogen sulfate was more successful than the other because they observe an increase of the shoot size and the root size of the tomato plants. So they selected this treatment for the biomass, and you can see here on your right hand side that indeed the tomato plant is looking taller and the roots are bigger. So we were happy to see that our consortia has a positive impact on the growth of plant size, and we are doing ongoing investigation to see if it can be used with other types of plants. So here we were really interested in our project to understand the relationship between algae and bacteria. And we realized that there is an exchange of metabolite that is happening at the level of the consortia. So the bacteria is releasing, for example, in our case, acetate that is used as carbon source by our algae, Chlamydomonas, and Chlamydomonas is releasing oxygen through photosynthesis that is very useful for the bacteria for its anaerobic metabolism. So we have a really symbiotic relationship happening, and depending on the condition and the wastewater that you are using, this relationship can be greatly improved or poorly happening. But from this investigation, we also wanted to ask the question of, is it possible to produce hydrogen, or at least produce more hydrogen in this case? We know that algae or Chlamydomonas is producing hydrogen. We've been working on that for many years. And we know that some bacteria do also. So would it be possible to improve this hydrogen production rate? And next would be, of course, to understand how this happened and how can we improve it. So the results were quite interesting because yes, we were able to improve the hydrogen production rate using our consortia. Here in blue, you can see that over 4 days, we can reach around 35 milliliter per liter of hydrogen using the Algae-bacteria consortia. And this is we confirmed also that the consortia is well, meaning that it's growing happily because the dry weight is greatly improved. We reach almost 12 grams almost 12 times more biomass than the control. And if we look in total hydrogen production over 30 days, in the bottom graphs, you can see that the consortia produce way more hydrogen than the controls. And similarly, the chlorophyll is higher, meaning that our algae is very happy in those conditions. So those results were obtained from the wastewater from rural areas, the same wastewater as before. So here we have quite a complete cycle where we are able to grow our consortia in wastewater, and then we produce hydrogen, and we are able to get biomass that is useful as biostimulant. So what is interesting here is to have an economic or energetic evaluation of the overall process. And we don't have those data because the project was only 2 years and it was at the fundamental level. But our next project is envisaging to do that. We are trying to upscale that process and to do an overall economic analysis of how we could use that in terms of biorefinery. Are we able to connect our process to commercialized application? Excuse me. So of course, when you want to commercialize things, you need to look at what's sellable, and so biostimulants is a great market. So that's one plus. But we are also interested in biofuel in general, because we need to transition to a greener society. And so we need to remove our fossil energy dependency by using other type of renewable energies, and hydrogen is one of them. Of course, we have biofuel in general, bioethanol, biodiesel, and it's been done for many years now using crops. But we know that there is a problem, because using crops, you use it either for biofuel or for food. So we need a system that allows us to remove this dependency. And algae or microalgae in general are perfect, because they do not require fertile land. They need more than 100 times less than plants, land than plants, and they do not increase the deforestation. And of course, they produce hydrogen. And as you saw just before, we are able to produce an interesting amount of biohydrogen, and we hope to be able to produce more. One thing very important is that hydrogen is 100% clean, because it does not release CO2 in the atmosphere. And actually the combustion produces water. So it's a very important energy sources. And it's something very important in Europe right now, because we have this Green Deal happening. And so it's this Green Deal is focusing on sustainability, on any kind of project dedicated to fighting climate change. And it has a specific call on hydrogen production, and meaning any type of hydrogen, and biohydrogen is included. So we are very happy, because hydrogen again is very energetic, and it's so energetic that you can use it for space rockets. So this demonstrates that basically we could use it for anything. And actually right now we know that we have cars being developed, being produced using a mix of hydrogen and electricity, because we cannot store electricity, but we can store hydrogen, and we can transform into electricity, and electricity back to hydrogen. So it's a very convenient system. And is not just something in the future, it's actually happening right now. We have a lot of hydrogen refueling stations in Europe. And actually, I had this slide for last year where it was saying that basically 20 station would be added in Spain. But since then, of course, we had the COVID. So it's a bit of a statu quo, but you can see that the map is quite developed already. So for sure, we will be using hydrogen in cars for everyone pretty soon. And you can see that the hydrogen future is very bright, because for the next until 2100, we forecast hydrogen as being the most important fuel. So I want to a little bit to introduce how hydrogen production is happening in the cell, because I think it's quite interesting to know how the cells, photosynthetic cell is able to do that. First of all, photosynthesis is able to allow the cell to fix CO2 and then to release sugars. And that's exactly what you can see here. When the light is entering the cell, photosynthesis is happening, meaning that you have electrons, thanks to a chemical reaction, the electrons that are released, and the cells is able to produce carbohydrate or sugar. But in another way or other conditions, the cells is able to release hydrogen. Unfortunately, at the same time, it's releasing oxygen, and oxygen inhibits the enzymes. So this is why actually hydrogen production is not that easy in reality, because it happens for a few seconds before it's inhibited. So in the lab, we need to find strategies and understand really how to prevent this. So you would consider the cell being a solar panel, and you have basically one protein here, ferredoxin, that decide that the electron needs to be used for growth. So that would mean biomass production. But sometimes those electrons are so many that the protein decide, okay, we cannot grow anymore and we have to produce hydrogen. And this is exactly what's happening in the cell. So now the electrons are going directly to the hydrogenases enzyme. Of course, the oxygen comes along and stop that enzyme production, and we need to understand how to stop that. But basically, you would consider this enzyme as this type of cooking instrument where it's releasing the pressure. When there's too many electrons in the cell cannot carry on growing and growing, otherwise it will die. So it decides to produce hydrogen to use some of the electrons. So we have two themes really of studying in the lab. We need to understand how the oxygen is blocking the enzyme, or how to stop it. And we need to understand how the electrons are divided to either growth or biomass, to hydrogen, or any other pathways that are dependent on this ferredoxin protein. So how do we do that? We try to do genetic modification since we have tools available to us. We try to modify the growth conditions, and we also search for new types of microalgae. So in the case of genetic modification, basically what we do is we try to stop this oxygen entering in the enzyme. So that would be modification that render the enzymes oxygen tolerant. We have cases, for example, with Clostridium, that show us that the enzyme is more oxygen tolerant and is producing more hydrogen in general. So it's very useful for us to see that it's working. But of course, when you have a mutant, how do you select it from the wild type? Well, you grow your colonies on the typical Petri dish, and then we have a chemochromic sensor that allows us to see if our mutant is producing more hydrogen, because this sensor is reacting to hydrogen and turning blue. So it's quite efficient, but it takes also a long time. It's not that quick, because you need to grow your cells first, and then you need to wait for the hydrogen turn into blue. Then you need to improve the growth conditions. And we know that clammy is not very happy, but accumulates more electrons when it's on diet. And that means removing sulfur, removing nitrogen, or removing phosphates. And with this condition, we know that it's producing more hydrogen. In our lab, we developed a new condition, which is low light, because low light produces or releases less oxygen, and we don't remove, we add nutrients such as acetic acid. And in our system, we grow the cells over a range of 5-6 days, where we have added acetic acid. We grow it in low light, meaning 20 microangstine. We open the top, so we have a little bit of oxygen, and we re-add acetic acid. So you see there's like a statical phase, but then it grows, hydrogen production improves again. And we are able to do that over many days, up to 30 days. But here you can see that after 12 days, we improve our hydrogen production rates 20 times compared to our control, which is quite low. So we have now a new method to improve hydrogen. And of course, the last way to get more hydrogen would be to be looking for a new strain of algae. And what's good about microalgae is that they are everywhere on Earth. So you can find them really in any continent. This is why the biorefinery concept is quite interesting, because it could be applied to anywhere, and it's so much everywhere that it's even in Antarctica, in the South Pole. You can find algae in the middle of the penguins, the seals, you name it, just right there. And you can find green and red algae there. For us, it's very interesting because they have different metabolism, and they are adapted to cold. So they have different types of enzymes, and they can survive in extreme environments. So the cells need to use their electrons in a different way, let's say, in order to survive and be able to metabolize and be well and alive in those conditions. So to go back to our algae, since they are everywhere, and with biomass we can do almost anything, they are really useful, and I really advocate for the development of microalgae in general, because we do any type of bioproduct using them. So, of course, we have to go back to this biorefinery concept. And now, we can add algae and bacteria. So we study that in our lab, but we are hoping that it will be developed in many labs, so we can find new conditions. We can select new algae strain or bacteria strain, and maybe get even better results. We want to really connect this growth to CO2 fixation to help us depollute a little bit the atmosphere, and use wastewater. So in our lab now we are using wastewater from dairy, and olive oil, which is an industry, big industry in the south of Spain. And we are looking at the same parameters, growth and nitrogen consumption. And then we are working with our partners to develop systems that produce even more biomass. For hydrogen production, unfortunately, we need to work in closed photobioreactors, because otherwise the hydrogen is released in the atmosphere. And we also need a system to collect it. But then the biomass obtained is harvested, and again, other technical solutions are being developed to make sure that they are cheap. So we are not spending even more money on developing those techniques to get the biomass, but also that they are very efficient, because we need to have a maximum of proteins, a maximum of lipids, and so on, to be used in end products that have high value. So in a biorefinery concept, what's important is to produce what's saleable, of course, but at the same time discover new range or new type of bioproducts, in order to have a diversified market. And I will stop my talk here, just to say that the biorefinery concepts can be done anywhere on the planet, as long as you have industry that are polluting with CO2 and releasing wastewater. That's why we need to enforce this kind of investigation and also application. Because also we have bacteria available anywhere. So that's it. Thank you. It's not that stable. It's just that we are able to grow it. But that doesn't mean that we don't have any other organism, okay? We start with a stable consortia in synthetic media, but once we move it to wastewater, it's still there, but we have other things. And the consortia level is decreasing, but we still have some. So it doesn't mean that it's really stable, but we still have traces of the consortia. What we want to do is transcriptomic to really see the ratio, but because we don't have an idea of the ratio of this consortia versus the other microorganisms, okay? So I don't want to say that it's just this consortia. We started from 4 because really it's the contaminants we got in the laboratory. We started having contamination and we thought, okay, why? And then, so we were trying to get rid of those contamination as much as possible. And then we thought, no, maybe it's just a natural partner for our algae. So we have, I think we had 2 or 3 from our lab, and then we had other labs around us that had contamination, and we asked them. The biohydrogen production rate is lower than any other techniques, but what's convenient is that we are using the photosynthetic machinery, so potentially we could improve by tenfold. If we improve by tenfold the photosynthesis, then we would have way more hydrogen. Of course, it's not that easy to improve photosynthesis. Solar panel, of course, don't produce hydrogen, but they are very efficient at producing energy. And so if we are able to improve photosynthesis, and so many labs are working on that, we are not, because we are not specialized in photosynthesis, but then we can improve hydrogen production because we might be able to get more electrons going directly to the enzyme. For us, we are more focused on the second half of hydrogen production, which is once the electrons are inside, so after the photosynthetic process. And we have a great enhancement of hydrogen production when the algae is combined with the bacteria, because now the bacteria is giving more carbon source to the algae, so that it's feeding the algae basically, and the algae is growing more and producing hydrogen. Although at one point it has to decide, I need to stop growing somehow to release the electron that it has. So this is also an important question. At one point, are we able to grow way more, and produce way more hydrogen? Is there a way to combine both traits so that we can have more biomass for biofertilization, for example? That's why I insist really on the biorefinery concept, because if you just look at hydrogen production, then it won't be something commercializable pretty soon. But if you integrate it into this biorefinery concept where you remediate wastewater, you use the CO2 from the atmosphere, you use the biomass you obtained, then if you produce hydrogen, this hydrogen can be recycled as energy sources for the whole biorefinery. So it could be in auto capacity, if you like. And then it can be a successful commercial application, because if maybe you don't use it directly for cars, right now because the ratio or the rate is not high enough. But in terms of the biorefinery system, it's really useful, and it will be a commercializable process.